Hello, students. Welcome to the second class of this course, Plant Biotechnology. And in this class, we will learn about molecular cloning. As you know, that for any gene manipulation experiments, the first thing is that you have to construct some vectors. Those vectors include, so basically, what is a vector? Vector is nothing but a piece of DNA which you can use for cloning of your gene of interest. And this vector has other features also, like it has origin of, it's basically plasmid vectors you must have heard of. So origin of replication is there, antibiotic selection marker is there. So these techniques actually, so molecular cloning basically the involves all those components. But we will, before we come to all the these vectors and what is the gene fragment or gene of interest or which you want to clone, let's understand the definition of molecular cloning. So it's a process by which recombinant DNA molecules are produced and transformed into a host organism where they are replicated. Here, the DNA fragment of interest is actually the target is it's more having more copies of the same gene of interest. And as I told you that a plasmid or a vector backbone that contains all the components of replication in the host, I have already explained. So you can now understand that there are two main components in the molecular cloning besides the reagents. So one is the gene of interest you are trying to clone, and then second is the vector. So this vector is also important. Now, first we will learn about the DNA of interest or gene of interest. So this gene of interest can be a piece of DNA fragment from either complete or partial genomic sequence, or alternatively it can also be complete coding sequence or a part of coding sequence. In promoter studies, promoter characterization studies, you can even clone promoter sequences also. So that can also be a part of can be a fragment of interest. And additionally there are other components which are also routinely cloned in biological experiments, like these small RNAs or long RNAs or long non-coding RNAs. So all these makes a part of molecular cloning. Now if you look at the characteristics of plasmids, then which we use it as vectors. So plasmids are nothing but extra chromosome molecules of DNA in bacteria, and their size vary from 1 kb to 200 kilobase pairs. They are double stranded circular molecules. As I told you, they are bacterial features, so they are found in a variety of bacteria. And most plasmids have narrow host range. That means they cannot survive in in some unknown host. Okay, but their replication is independent of the replication of bacterial chromosome. And also it's the number of plasmids in a bacteria varies for different plasmids. There are some plasmids which are are high copy number. There are some plasmids which are low copy number. And so I told you that there should be origin of replication, antibiotic selection marker gene, and to clone a gene fragment, plasmid should also contain a multiple cloning site. So these are the features of uh, of vector. So we will stop here, and in my next class we will talk about the agrobacterium. Don't forget to join my next class.